Hey everyone, it's a dose of Dr. Drew.
So, little technical problems today. Are you guys hearing me okay presently?
Everything's going haywire.
Let's just make sure that we hear the sound. Looks like they're not hearing me either.
Well, there's a delay, baby.
We'll see. Susan and Jeans, very good. Let's see, give you guys a second to line up.
Yes, it's good.
All right,
okay,
good.
thank you, Stephanie. And who's your lady?
All right, excellent, guys. Thank you for being here. We appreciate it. We have a very special guest today. I'm going to get right to him. I'm not going to spend any time answering questions.
Hold on a second. Let me set that up now.
or going over the data because we have somebody who really knows a lot about this. He's a scientist who studies COVID-19.
Wait, hold on. Don't get excited yet.
We're having technical problems. We
I
can't
can find him.
Uh-oh.
Oh.
I'm here, I promise.
Yeah, I know.
He's
I
here.
hear him.
Okay.
We're going to get his face out here.
All right,
before
hold
he
on.
does.
I
So
promise.
let me finish the intro, Susan. You just be quiet about whatever it is you're doing. And here we go. It's a scientist who studies COVID-19, and he does a daily update on TikTok. And I watch his updates on a regular basis, and I learned something from them, and I thought, we should interview Dr. Nock, Dr. N-O-C. That's what he is on TikTok and Instagram. You can find him there. You'll recognize him when Susan brings him up here quietly as she is working. There he is. Hey, Dr. Nock, Morgan McSweeney. Thank you for joining us here.
Dr. Drew, pleasure to be here. Thanks for having me.
It's really a pleasure. So one thing I don't know about you is what your training and background is. I know you're a virologist or you're a bench scientist of some type, but tell me about your training. Where'd you go to college? Where'd you go to grad school? Specialization, postdoc, all that stuff. Let's hear it.
Yeah, that's a great question. It's hard to fit, TikTok videos 1 minute. You can say, I'm a scientist who studies COVID-19.
That's enough.
You get 3 seconds.
You got my attention with good information after that.
Yeah, that's it. So for my undergrad, I went to the University of Central Florida, studied biomedical sciences, which is sort of a catch-all: molecular biology, microbiology, viruses, humans, lump them all in together. Decided I really liked doing research, so I went to grad school at the University of North Carolina at Chapel Hill. It was under the broad term pharmaceutical sciences, but I was really focused on immunology and how that relates to pharmaceutical sciences. I was looking at unwanted types of immune responses. So, if you have an immune response against drugs, like therapeutics, that's a bad thing because you eliminate those drugs, they don't work anymore, you're at increased risk of allergic responses. And so we were working on ways to basically prevent those from happening. Got my PhD and now working in relatively small biotech in the development of drugs to treat respiratory viruses. So even prior to the pandemic, I was working on antibody-based drugs for things like influenza, a couple other respiratory diseases. And then so now just sliding into that pipeline, we're working on COVID-19.
So monoclonal or polyclonal antibodies against viruses, would that be a summary?
That's correct. Yep, monoclonal antibodies.
So I don't know if you're aware, I'm just getting over COVID. I had 10 weeks of hell. And I took bamlanivimab, which is the Lilly monoclonal antibody, and it kept me out of the hospital. There's absolutely no doubt in my mind that is what turned me around. Susan, I have frozen on the screen here. I don't know if you're aware of that. Dr. Nock is still Dr. Nock is still fine, so I'm going to keep talking.
I see you frozen too.
Gosh.
Yeah. It's all right. They don't need to see me.
Oh my God, but that's not a good frozen.
So are you coming up with some newer? Now you took Dr. Nock off the screen too.
Okay,
Susan.
let's see what they say on that.
There you go. So are you coming up with similar kinds of products?
Very similar. And so bamlanivimab is a systemically administered monoclonal antibody. What it's really good for is for keeping people out of the hospital. Our goal is pretty much the same. That's pretty much across the board. Is if you give it too late, the virus has already had time to cause this sort of feed-forward cycle of inflammation. And then even if you take out the virus, you still have that inflammation, which is what's causing damage.
Yeah.
So, yeah.
So I talked to a virologist, a guy named Larry Brilliant, and he made an interesting observation. He has seen a lot of smallpox. And he said, clinically, although we're very focused on this being a respiratory virus from the standpoint of lungs being prominently involved, transmitted via nasal pharynx, he goes, but when I really look at the clinical picture, it reminds me more of smallpox. And I thought, boy, that's an interesting kind of way of thinking of this illness. I never would have done that. Do you agree with that, or is it a bridge too far, given that most of what he's talking about as smallpox is really a multi-organ problem from the inflammatory response?
Right. I think the latter. And so this has caused a lot of confusion in general, even if you think about people who say, I already had COVID back in back in November of 2019 or something. The issue is there's a lot of diseases that cause really very similar sort of phenotypic, what it causes to the body. And then the actual viral agent can be totally different in terms of how it's acting, the types of drugs that would work on treating it. So I don't actually know too much about the clinical presentation of smallpox, but my guess is that it's just the body may be reacting similar, even if they're totally different.
Got it. So it literally is, strictly speaking, the clinical course, in the sense that multiple organs are involved, multiple organs are failing, not the mechanism isn't similar in any way.
Exactly. Yeah. And so that's where, SARS-CoV-2, the virus that causes COVID-19, it's entering cells through that ACE2 receptor. And one of the things that's unique about SARS-CoV-2 is that's not just in your lungs. It's not just in your respiratory tract. You've got that ACE2 molecule, that sort of key, that lock that it can get into all throughout your body. And so that's part of the reason we think why you're seeing not just respiratory effects from COVID infections, you're seeing these systemic types of effects, which maybe increases the risk for these lasting types of symptoms you have.
Anything special about the brain as it pertains to an ACE2?
There may be, not that I know of. I don't really know.
Because I feel like my experience with the virus kind of feels like that was a prominent area of infection.
Right.
And now it might have been the inflammatory piece. I mean, subjectively,
Right.
I just couldn't tell.
Yeah, that's what I was going to say. Is we do know that there can be vascular components to disease, and so if there's any cerebrovascular impact of the infection, it's causing, even if it's not acting on the brain cells themselves, if it's acting on the vasculature in the brain, maybe there's something there. Right. I don't know. I'm making that up though.
Right. I get that. I think Susan, am I looking at the right camera here? She's been flipping cameras all over the place.
The big camera, the Ellen froze.
Got
I'm
it.
using the backup now. Look at me.
Okay.
Okay, there you go. You're both together now.
All right. So I suspect it's going to be all of the above when we get back. We're going to get down to it because it's, there's I've read some literature that shows there's microvascular damage to the olfactory nerve as the source of the loss of smell. I had really a significant problem in my 8th cranial nerve, my ear. So I'm suspicious the same thing went on there, although it could be in my temporal lobe, who knows. And I had a significant long hauler inflammatory response. So it all kind of seemed to be going together, both viral infection, microvascular damage, and inflammatory response.
Yeah. But one of the things that people are really interested in is if you get the vaccine, in addition to reducing your risk of being hospitalized, is it going to reduce your risk of sort of these long hauler symptoms? And I really do think it will, for a couple of reasons. One, just broadly, we know the vaccines overall are going to reduce your risk of being infected. We didn't know that at first, but now there's increasing data that the vaccines do help reduce your risk even of like an asymptomatic infection and transmission.
What's your hunch on how strong that effect is?
Yeah, so the first numbers that are coming out are going to be from when the vaccines were sort of battling against the non-variants. And those numbers are sort of in the range of 60, 70-ish%.
Okay.
My hunch is that that's going to go down.
Okay.
As the variants gain sort of market share within the population. It's not going to go to zero. Who knows where it'll end up, whether it's 30, 40, 50%, but it's still going to help reduce your risk of infection overall. And you can think of this sort of in tiers of challenges for the vaccine. The hardest thing to do is totally prevent any infection.
Right.
The next level up is you still get infected, but it's only a mild disease. And then the last level is you get infected, maybe it's a mild disease, but you're not hospitalized or you don't die from COVID-19. And so at those different levels of difficulty, you see corresponding differences in sort of vaccine efficacy, where it may give you a 40% reduction in any infection. It may give you a 70% reduction in mild disease, and it may give you an 85% reduction in hospitalization.
Yeah, this is where people get confused. As soon as we start talking about these different levels of protection for particular clinical syndromes, people's eyes glaze over very readily. Let me. You
can't compare them directly.
Yeah. So the 70% asymptomatic prevention of asymptomatic spreading. Then we have, if I'm reading this data right, is it not the case that Johnson & Johnson had about a 65% ability to fight off the variants in terms of infectivity? Did I get that right?
That's correct. Yep.
Okay. So Johnson & Johnson is showing some, and by the way, in the range of the flu vaccine, which is 70% effective, right? So
yeah, sometimes on good years.
Yeah. So the fact that we get 70% protection measured against the variant, I'm as a COVID survivor, I'm kind of thinking, well, that's the thing I want protection against. Maybe I should be taking the Johnson & Johnson vaccine. Am I thinking it through properly?
It's a good result. It's hard to compare.
Yeah.
And sort of Moderna and Pfizer were tested before the variants were around.
Yeah.
Just because they haven't been tested against the variants, though, they're still going to work against the variants. I'm pretty sure. We
It's
just don't know
about the same
exactly what
level.
hit they're going to take.
Right. About the same level, probably, right?
Right.
Yeah.
And so we do know that Johnson & Johnson has been tested in South Africa, where 95+% of the cases that they saw in the clinical trial were with sort of the most scary variant we see, which is
Right.
B.1.351.
Yes.
So, the numbers are lower, but it's a harder battle for that vaccine. So you can't compare it directly to the Moderna. In some sense, it's been validated. It's been validated against the variants. So
Right.
it's pretty good.
Right. That's what I took away from the J&J studies. It was 100% hospitalization. No, 100% against death, right? It was
And hospitalization in the trial.
and hospitalization. Was it 80% against moderate COVID, something like that? Yeah,
It was 85%.
Yeah,
for
and then
severe.
60 something against variants. I'm like, well, that sounds pretty good to me. I think I want that vaccine, especially. Now this is my other. Well, let me finish my range of thought here before I get into my own personal thing. You mentioned mild disease, that if you get with the vaccine, we expect if you get infected at all, you're probably going to get mild disease. Is that a fair statement?
Correct. Yep. Even if you still get infected, your risk of severe is decreased.
Right. And even if you get mild disease, that doesn't mean you're infectious. So we haven't answered that question yet, correct? Now, like, for instance, myself, I only spread to one person, and I had moderate to severe disease. Just because you get the disease doesn't mean you're infectious, correct?
That's correct. Big difference there. And so the biggest difference is if it prevents you, if you would have had mild disease without the vaccine. Now, with the vaccine, you still get infected, but it's asymptomatic, totally asymptomatic.
Yeah.
We know that you exhale fewer viruses if you're totally asymptomatic compared to if you are symptomatic.
Right.
And so I would expect that extra boost of immune protection you have, that gives your immune system a head start. You're shedding fewer viruses. My guess is there will be generally slightly lower levels of transmission. We don't really have those data yet, but we do know it's sort of improving across the board.
Right. And I'm seeing a lot of data come in from Israel. Is that what you're talking about?
Yeah, they sort of are leading the pack in terms of rollout, and correspondingly, a couple months later, we're starting to see the real-world impacts where, they've been using mostly the mRNA vaccines. I expect to see similar levels of correspondence between clinical trials of all the vaccines and the real-world rollout. You take a slight hit. In the clinical trial, it's sort of the perfect conditions. You've got the hand-selected people with the perfect cold chain, the perfect clinical follow-up and care. And so clinical trial may post like 95% efficacy.
Right.
If you go to the real-world study and then you still see 90% or 85% or whatever, that's pretty ideal. You're never going to hit exactly the same as those perfect conditions. But what we're seeing out of Israel is it's really close. And so that's really reassuring.
Yeah. And one of the things I want to at some point talk to you about is how do we emerge from this? How do we know when we're ready to emerge and that kind of thing? But before we get there, I have two other questions. So I got, very rapid onset, moderately severe COVID, and my wife was sleeping next to me the night I became ill with fever and chills and the whole deal. She didn't get this thing.
Pretty common.
Yeah. Talk about that. Do we know what that is yet?
We do. And so even from, very beginning when it was predominantly in China, people were doing studies on the risk of intra-household transmission. And the numbers are, you may think it's like 80% or something. That's definitely not the case. It's closer to 10 to 20%. If there's someone in the household, there's 10 to 20% chance that someone else will get it. And that's sort of dictated by two things. One is there's a lot of variability across infected people in terms of just how much virus they're putting out. Some people are putting out huge amounts of virus. Some people are not. You can't really tell the difference based on their symptoms. And most people are not. Overall in the population, it seems like about 15-ish, 20% of people cause somewhere in the range of 80% of new infections. And so in a household context, that means most of the time it's not going to transmit. And then on the second layer on top of that is there's behavioral differences.
Right.
If you're sharing a spoon, you're sharing a glass of water, that obviously increases your risk tremendously.
Someone is asking on the Restream, I'm going to read his question to you. J&J vaccine is more effective than mRNA vaccine for variants. What's the justification for the statement?
No, I don't think we did say that. I think we said it's been tested against the variants. So we have data.
That's right. We have data against
the mRNA
them.
ones haven't been tested yet. I would expect they're going to take a hit. We don't know how big that hit's going to be.
But you said, what I heard you say was you expect it to be in the range of the J&J vaccine.
Right. In that, it could go from 95% efficacy against not the variants. Maybe it'll go to 75%. If I was to guess, those are just random numbers.
Right.
It's going to go down somewhat. It's not going to go down to zero. It's sort of what I'm trying to say.
Do you have any preference which vaccine you would take?
I would take literally I haven't been vaccinated yet, by the way. If someone showed up at my door right now with any of them, I would take it immediately.
That's kind of, yeah, that's what I think that's a smart way to approach it. I originally was sort of had my eye on the Johnson & Johnson because it feels like a more traditional platform. Am I kidding myself?
No. Yeah. So the adenovirus platform, it's been used in an Ebola vaccine. It's been used in a couple other investigational ones. The mRNA are new. That being said, I'm not scared of the mRNA ones. Right. If I was offered an mRNA first, I would equally take that.
Right. And certainly now with millions and millions of people vaccinated, it's looking pretty good.
Yeah. And that's the thing is most of the adverse reactions that would happen after vaccination, pretty quickly.
Right.
Anaphylaxis, allergic responses, those happen very quickly. The other types of things that could feasibly happen within a few months. And so, we sort of know that they're working really well and they're pretty safe.
And people are focusing. I read some of I saw some people publish some of the untoward effects, and they all were just general medical. First of all, a lot of them seem very mild, and the ones that seemed more serious were things that we see every day in the medical clinic. I mean, things that happen randomly.
Right. Yeah, and that's the big challenge, and that's the big, the VAERS database is a source of a lot of confusion. When you vaccinate 75 million people, you're going to have some bad clinical outcomes that happen very shortly after vaccination.
Yep.
And a lot of people say that sounds a lot like the arguments, that people were making about COVID. Oh, people got COVID and then they died. This is a very different circumstance because, you can draw a direct link between the pathology, the viral infection, the inflammation, the radiology results, and you can say very clearly these things were caused because of the infection.
Right.
Totally different story. Someone gets vaccinated. There's no mechanistic clinical explanation for how it could cause some of these things. And most importantly, these things are not happening at a rate above what you would expect sort of in the general population.
Right.
In contrast with COVID, you see the excess mortality charts, which is where surprisingly, you can actually predict sort of the total number of people that are going to die basically any week of the year. It's pretty reproducible.
Yeah.
And so you can plot that and you can say, okay, if it's more than this much above those expected levels, that means something out of the ordinary is happening. And so if you look back to the pandemic, you see this huge spike in total excess cases, which we're not seeing with the vaccines,
right.
which is a good thing.
Right. And there was kind of an interesting thing that happened in the first 6 months. You see this or 9 months, and then you saw it drop down briefly below normal, meaning that a lot of people that died in that group were going to have died in that next year anyway.
Yeah, I don't know if we ever got quite, now we're starting to come back down to the expected range. I don't think we ever actually dipped below sort of the predicted value. And so I wouldn't quite say, these people died 2 months earlier than they would have otherwise.
Got it. So let me talk about my own sort of clinical situation. So I have these I've been having a antibody profile done, and I have antibodies against 5 different antigens, nuclear capsid proteins, spike proteins. I have all kinds of stuff going on, and they measured me against sort of an average vaccinated clinical spectrum, and I'm like 10 times above vaccine level. So I figure, I'm going to bet that could put me at risk for a pretty nasty reaction when if I were to get the vaccine. B, I don't want to take the vaccine from someone who needs it. C, with all that antibody to multiple different antigens, why would I need a vaccine at this point? Do you agree with that?
I agree with B out of A, B, and C. And so A is also valid. What I do know is from population-based studies, you can compare on average people who get vaccinated, the titers of antibodies they have against the titers of antibodies the people who have recovered from disease.
Yeah.
They're usually at least equal. Sometimes the vaccinated is a little bit higher.
Yeah.
So if you happen to be 10-fold higher than the vaccines, that's excellent. So B is an important thing. There's a lot of confusion about this.
Yeah.
People have recommended you wait 3 months after you've been infected to get a COVID vaccine.
Right.
That's not necessarily due to any safety considerations. That's mostly because you probably are pretty well protected, at least for a few months after your infection.
Right.
And so, that dose could be routed to someone who doesn't have one.
And to be fair, maybe a few years. Right?
Yeah, certainly.
Shouldn't we be measuring antibodies on people just to do it? I mean, if we can, if there's resources to do it. Okay.
Well, the hard part is it's hard to draw a correlation between you have X number of antibodies and you're protected. We don't have that threshold yet.
Right. I've been using other vaccinated patients as my threshold. If I drop
and
down
that's pretty
below,
safe.
yeah, if I drop down below average vaccine level, then I'm going to get the vaccine. Then it's time.
Yeah, I would say there's probably no detriment. You get the booster shot. What can also change is the quality of your antibodies, which is really hard to capture and sort of, if you're just looking at the number. We know that from your primary shot of your vaccine, you have a pretty good total antibody response. When you get that second dose, what happens though, is you have 10-fold more neutralizing antibodies. And so those are the ones that really stop the virus from getting into your cells because they're blocking that spike protein.
Yeah.
And so if you're just looking at total numbers, it can be difficult to say, are they the neutralizing antibodies or not the neutralizing antibodies? And getting that booster shot, where you can think of your first dose of the vaccine after you've been infected sort of as a booster, that's going to give you those really high quality antibodies.
I had 95% neutralizing antibodies. Pretty good, right?
That's fantastic.
Yeah, it's ridiculous.
So realistically, I would expect protection to be, I wouldn't expect people from the clinical trials to start getting infected before 6 months, before 8 months. We know from other coronaviruses, you can look at people's antibody titers over time against the seasonal coronaviruses. And you see it spike, basically once every 12 months or once every 24 months or something, which suggests that immunity sort of broadly, at least against a couple other coronaviruses, you don't get infected every other month. It's usually, at least about a year. We don't know exactly how that's going to compare to this coronavirus, but I would be pretty surprised if you had to get, vaccine booster shots more frequently than a year.
That makes sense. See. I'm trying to pull up my vaccine profile here so I can tell you all the different proteins and the numbers and stuff. But it's a pretty sophisticated panel. This is not just what you get from Quest Lab. You know what I mean? This is not just going out and getting a sort of loosely quantitative analysis. Well, I'm not going to well, maybe this is it. Maybe this is it. Is this? No. Oh, yeah. Oh, it's locked. What is my, hold on. Give me a second.
You're going to flex on us with your antibodies?
That's right. I'm going to spill them all over everybody. So what do you feel about convalescent antibodies now, convalescent plasma?
The data have been not as convincing as I would have hoped.
That's what I'm saying.
It looks like you have to use it early, and you have to use it with where the plasma has very high titer antibodies.
So you're much better off.
It can help a bit.
Much better off with your monoclonal antibodies that you're producing.
Yeah. Although, so the one difficulty is monoclonal antibodies. It's just it's targeting one target on the virus, which makes it super susceptible. If there's a mutation right there at that one spot it's targeting, you can see a 10-fold drop in neutralization potency or a 20-fold drop, which you don't really see with sort of a vaccine response, or you don't see with convalescent plasma, because there your immune system's attacking every different angle of the spike. And so even if one of them is mutated, you still have attacking all the other angles. And so that's why we see like a 3-fold drop in neutralization potency with sort of vaccine serum, but like a 10, 20, 30-fold drop with some of the monoclonals. Some of the monoclonals, though, are now being developed specifically to address the variants. Those are still okay sort of for now. We see more.
Right. So it's evolution. People, if any of you out there doubt evolution, just watch COVID-19. just watch coronavirus 2. Real-time evolution. That's what you're watching. That's how biology works, everybody. That's how it works. So I have anti-SARS RBD IgG at 105,000. I have anti-SARS S1 IgG 71,000. I have a nuclear protein NP IgG 77,000. Those are insane levels.
Great. I wish I'm at 0.
And I have IgM and IgA also coming on board, coming down the line, as we say here. So yeah, so I don't think I should take a vaccine. I think other people need it more than me.
Yeah. For the second part, from a selfish perspective, there's no harm for you getting one. It would probably give you even more neutralizing antibodies. But since there's a lot of people who haven't had one at all, they could certainly benefit more than you.
Well, we have maybe some international travel coming up, and they may force me to get a vaccine for that travel, and that's probably when I'll. In the meantime, I'm just going to follow my antibodies. I want to be a subject where people can. Let's get the time course of these antibody responses. Let's see if they decay. Let's see. I want to check my cellular immunity, not just my humoral immunity. That it seems to me the cellular immunity story hasn't even been told yet, has it?
It's ignored. It's harder to measure.
Yeah.
And so the T cells, people think about the antibodies because they're good. They can totally prevent an infection if they're in the mucus. The T cells, though, they play an equally important part. They can be equally predictive of how protected you are against severe disease, but it's a lot harder to run those assays. It's a lot more expensive to run those assays. So the data are much more scarce.
So I'm answering somebody on the thread who, Leopold, was worried that somehow the vaccine would confuse our own or suppress our own antibody response. And what Dr. Nock is telling us, no, That would not happen. It would just improve or expand our profile, which I like that. I like that. Somebody's saying I'm good for at least 6 months with my antibodies. That's probably right, wouldn't you say?
I would say so.
Yeah, I would too.
You know, we don't really have the data yet from the clinical trials. I would be surprised if it was shorter than 6 months, though, for sure.
Yeah, me too. So children, are we eventually going to vaccinate children, do you think?
I think so. I think we'll get to that point. We'll have the data from the trials in kids now. They're ongoing with ages 12 plus. We'll probably have that by the fall, I would guess. So, you know, sort of those ages should be vaccinated in the fall. Younger, I would anticipate probably not prior to the spring. We'll eventually get there. It just probably not for a little while. Luckily, children obviously are at way decreased risk of severe disease. That's not to say that they don't get severe disease. Very young kids have died from COVID-19. Kids have recovered from COVID-19, gone on to develop that inflammatory disease, MIS-C. So they'll certainly benefit from a vaccine, no doubt.
Now, Matt Mizu, there is nothing that I get. Where am I looking, Susan? Over here. There's nothing that I get that is because of any super insurance plan. Nothing. Bamlanivimab, covered even by Medi-Cal. The government bought it all. So it's covered for everyone. The Additech score, everyone can get my antibody profile. I don't know what the price point is yet, but I'm told by the company it will be quite affordable. Everyone can get these things. You just have to know that you have to ask for it or know where to go to get these things. The fact that doctors weren't using Bamlanivimab was shocking to me. That we could have reduced the hospitalization rate rather dramatically if it were used. There were 600,000 doses sitting on the shelf in Bethesda or something that could have been 600,000 people that stayed out of the hospital essentially for free. They come to your home, they administer it, there's nothing to it. I'm hearing lots more, Morgan, also stories of people who are now that they finally started using the monoclonal antibodies a little bit more briskly. People are reporting stories that of what I experienced was I actually felt better during the infusion. My infusion nurse said, "Oh yeah, I hear that all day," and now I'm hearing it from multiple sources. So isn't that interesting?
You mean like for the couple hours when you're actually being infused?
It's an hour infusion, and 30 minutes in, literally colors got brighter, and I felt stronger. And he goes, "Yeah,
I've not heard that."
Yeah, no. Apparently, this infusion nurse told me. He goes, "Yeah, all day long." People tell me that. I've heard that from other nurses that are doing the infusions as well.
Okay, yeah. So the thing to compare, see how the people are feeling who are getting infused with the saline, and see if they
right.
start seeing the colors.
I'm with you. In fact, I was worried about a placebo effect too when I was telling my nurse. I go, "Look, I'm just going to report this to you just in case." And
sure.
And what he said back to me was, "People tell me that all day long." Not that I'm just reporting, but that I'm stronger, my colors are bright. All this kind of very positive effect during the infusion.
Yeah, and to some extent, at that point, you don't care too much. If you're talking about reduced pain or reduced discomfort or something, even if it is a placebo at that point, you don't care as long as it's also actually doing the evidence-based clearance of the virus.
That's right.
If you feel better too, well, why not?
Do you want to tell them where to get the immunity test?
Additechscore.com, but I don't think there it is. I don't think it's available yet. They keep telling me it's coming, but it's almost here. They have to be geared up to be able to do a large scale, production of this scale, which again, I'm sort of advocating it as a more sophisticated way to approach. I appreciate what Dr. Fauci said, like, okay, after, generally speaking, after 90 days, it's probably time to get the vaccine, but there are more sophisticated way to do this. And I was talking to some cruise officials too. If you're going to get on a cruise ship, hey, this is a way to manage the risk of the population you're bringing on board. If everybody's like me, yeah, sure, test the rapid antigen test, but you don't have to worry very much about anybody getting the virus on board the ship.
Right. Yeah. No, if everyone has sky high antibody levels, they've got a lot of neutralizing antibodies, that's pretty much ideal. Luckily, we're getting to the point, or we will be getting to the point as we get into the summer here, where we're not going to be as limited, at least in the United States, in terms of vaccine availability. And so at that point, you may not even wait full 3 months after your first infection.
Right.
You may get infected, get your vaccine at 2 months or something. And then instead of 100,000, who knows how many antibodies you have. But once we're no longer sort of limited by vaccine supply, we're going to be in a really good place. I'm actually very optimistic.
Yeah, I think we're
going to start seeing some real progress here as we get through the summer.
What did your update say today on TikTok?
Today we were talking about, I haven't actually published it yet. Merck it's joining with Johnson & Johnson to produce more doses. This is the adenovirus vaccine. That's harder to do with the mRNA vaccines. Talked about that the other day. You can't just have a bunch of companies sign up and make more of the mRNA vaccine. A lot of people think it's purely that they haven't licensed the patent out, but it's just a really hard process to do. There's not that capacity.
You have said that the hard part is the lipoid membrane.
That's right. Putting things into a liposome, which is that little sphere of lipids that protects the mRNA till it gets into your cells, that's a hard task. It's hard to do it reproducibly. It's hard to get an efficient loading of the mRNA into those liposomes. And they work really well. There's a bunch of other liposomes, some cancer drugs. They're just hard to make. Unfortunately, it's sort of like one of the more difficult types of drug formulations. It's those are the vaccines that came to market first. So maybe it'll have some benefits for other liposomal drugs and other mRNA vaccines as we move forward, now that they've kind of been brought to the spotlight by this. But right now, it's a real challenge.
So you can't just ramp up liposomal production. That takes years.
You have to basically build the new machinery. You can't call up a contract organization and say, can we borrow your big facility that you already have? You have to build it. Right.
So here's a great question. You're the perfect person to answer this. And by the way, Matt Mizzou, the ICD-10 code is just COVID-19. Spike proteins also contain syncytin homologous proteins, which are essential.
I love this question.
Yeah, I do too. Essential for the formation of placenta in mammals such as humans. Isn't it possible the vaccine could stop placental development or has a cross-reactivity? What's the biology there?
Vastly misunderstood. And I have struggled for weeks and weeks to fit this into a 1 minute video. But
by the way, how long does it take you to make those? How long does it take you to make those 1 minute videos?
At least an hour.
That's what it's not 1 minute. Is the point? It was not a minute. Yeah, that's what I figured.
Not even close.
Yeah.
There's a lot of pre-writing and editing and everything. Okay, but syncytin 1. So this very difficult rumor started on an internet board, someone looking at the amino acid sequences between this placental protein and the spike protein from the virus. And so the thing to understand about proteins is they're not in a linear, it's not a chain of amino acids. They're folded into these 3D conformations. And so what that means is you can't necessarily look at short stretches of the linear structure and use it to predict very easily the tertiary structure.
Okay, so explain what you mean to people by that, because I know what you mean, but explain what you mean.
Yeah.
That proteins have different
kinds. They take the sequence.
Right.
You can take a protein, you can string it out.
You can pull it out, right? You make it a string.
Right. That can give you the sequence of the protein.
Right.
But if you let it fold, as it does in biology, that long string turns into this big sort of knobby actual structure. And so the immune system recognizes that big knobby structure when it's looking at targets. And so what people have done with this sort of fertility question is they've looked at the linear sequence of syncytin-1, and they've looked at the linear sequence of the spike protein. And they see a few regions where there's the same order of amino acids in this one and the same order in this one. But that's basically meaningless. If you talk to any immunologist, any structural biologist.
I want to say this in the strongest terms. If you don't understand how a sequence is different than a folded protein, go back to the first and second law of thermodynamics now. You don't understand biology, because that is one of the first principles of protein biology in terms of where we end up with these proteins. It's so much of biology is the first and second law of thermodynamics. But if you don't get that, you do not get what we're talking about.
And so, you can find very similar levels of shared linear amino acid sequence between the spike protein and not just this placental protein, between collagen, between hemoglobin, between myosin in the body.
That's right.
And what we're not seeing is like vast autoimmune disease as a result of vaccinations or as a result of infections. There's a spike protein there. You would expect to see a similar issue if it was an issue. It's not an issue.
Right.
There's
not
no
an issue.
Right.
There's no evidence. Yeah, exactly. Yeah. Miscarriages happen. Well, who should pregnant women be vaccinated routinely, do you think?
Yeah, this is a context-dependent question.
Yeah.
I err on the side of yes, if you have general exposure to potential COVID. One thing we do know is if you are pregnant, you're at increased risk of more severe COVID outcomes. That can be bad for you. That can be bad for the baby.
More morbidity, but less mortality. Would that be correct?
I think it's more of
both,
but I would have to look.
All right.
Not 100% sure.
Okay. So hang on, had another good question here. Oh yeah, if the incidence is under 30, if the incidence of morbidity and mortality under 30 is so low, why do we aim to vaccinate? I have my own reason for wanting to do it. What is yours?
Yeah. Well, I mean, that's part of the reason why they're going to be last, under 30. And so any decision you make in the pharmaceutical sort of landscape, everything is a benefit to risk ratio. And so what you're saying in that question is basically the benefits seem low.
Yeah.
However low the benefits seem, the risks are way, way, way orders of magnitude lower.
Right.
So even if you think you've got a 99.9% chance of survival from COVID, and that's not even considering the potential for long-term, you survive, you get long-term effects.
Right.
99.9%, that's 1 in 1,000. When you look at the vaccine safety, it's way, way better than 1 in 1,000. We vaccinated 75 million people with no notable effects. And so the benefits, even though they're lesser in that younger, lower risk population, they still far outweigh the risks. But that's why they're last in line.
I would also argue is we have got to suppress viral replication, the activity of this virus, so we don't have such evolution in real time where the variants are showing up all over the place. And
that's crucial, for herd immunity.
Herd immunity to me is one thing, but but actually suppressing the replication, I think, has to be a separate goal to prevent variants. Because if we had some sort of terrible variant evolve, we're start all over again. Now, I think we're positioned to respond to it, but it could be a mess. And I just think that possibility to me leads me to want to vaccinate people at least down to like 16. Do you agree?
I mean, essentially everyone. You're totally right. Every infection is a chance. It's a really low chance, but it's a chance that a mutation is going to happen. And so you don't have to get all the way to herd immunity to start seeing the benefit of fewer infections in the population, means lower chance of mutations. And this is exactly the same reason why they're still recommending, even though vaccines are going out pretty quickly now, we still want to wear masks. We still want to distance, at least for the short time frame here, to cut down on as many infections as we can so that we get to that end line. The end line's in sight. We want to get to where everyone's vaccinated before we have another bad mutation that happens.
Right. I mean, the virus will be endemic. You agree with that?
Yeah. I don't think we're going to totally eliminate it anytime soon.
Right. Do you have any hunch what the incidence is likely to be, say, per 100,000 once we stabilize?
No idea.
Yeah,
Some
I don't think anybody does.
Epidemiologists may, but I don't.
I don't think anybody does, and that's why I worry about these school openings being tagged to particular 7 cases per 100,000, all this stuff. That may never happen, literally never happen, and that's very, very concerning to me. Oh shoot, I had a rush of other questions I wanted to ask you. Let me go to the restream here and see if other people have ideas. Sorry guys, I've got to read these questions quickly. All right, let's you may not be comfortable even speculating about this. Oh, I know what I wanted to say. The mutations. My understanding is most of the serious ones are translocations and have occurred in multiple sites simultaneously. There's something about that.
They've independently evolved in different geographic.
But the same mutation, and the mutations specifically were translocations. Talk about that if you can, because I didn't know what to do with that myself.
Yeah. On the one hand, it's kind of good news in that sort of the worst mutations that we think possibly could happen, we've seen multiple times pop up. And so on the one hand, we know we've sort of tested the landscape of mutations that could possibly happen. The ones that are giving the virus the most advantage have happened. And even against those, we know the vaccines are performing at least relatively well. Maybe we can update them. But what we are seeing is, the virus has a spike protein. It's got a special ridge in that spike protein. And that ridge happens to be very important for how it binds to your cells. And so mutations that are in that ridge are the ones that are standing out the most. The E484K mutation, that's the one that sort of dominates that South Africa reduced immune response. That one has developed in a couple different places around the world.
And that's the B.135.
Yeah, B1351 has that E484K mutation. One of the variants from New York also has that E484K mutation. And the vaccines still appear to prevent or, greatly reduce your risk of really severe disease, even with that mutation. Moderna just made a new version of their vaccine. Actually, they just shipped it to the NIH for clinical testing that has that new mutation in it. And so maybe that'll be better than the current vaccines. Maybe it won't. We'll see. But at least the current ones, they still help a lot, even if it's reduced compared to the old variants.
It's one of the reasons I lean a little towards that Moderna vaccine, because I know there's a booster coming, and I don't want a variant. I don't want the variant. Thank you. So back to the original virus. Again, I don't know if you want to comment on any of this, but there was lots of, let's call it rumor, about the nature of the structure of the virus being some, fingerprints on it that are common for laboratory manipulations and all this kind of stuff. Is that something you have an opinion about?
No, I wouldn't say I'm enough of a molecular biologist to sort of give an expert opinion on that. What I haven't seen is a collective opinion among molecular biologists who are convinced by those claims. You see a blog post here, an opinion article there.
Yeah.
But for the most part, my impression is that there's not any convincing genomic evidence that someone used a restriction enzyme and you can say, aha, therefore it was in a lab or something.
There was some, I forget, I read it once, but it sounded interesting, as though there was some sequence that's a common insertion, I guess using restriction enzymes or something to insert, or CRISPR, I don't know what they're using. But there's sort of a hallmark kind of an insertion. But, just because we do it doesn't mean that nature can't do it, right?
That's a great point. It could easily randomly happen. And the genomes are sufficiently similar to ones we see circulating in nature. My impression is that if there was very clear evidence, there's a lot of really good molecular biologists in the world. They would kind of collectively recognize that. It wouldn't be one guy or one lady,
right,
sitting there claiming truth and everyone else hiding.
Right. There'd be a consensus. All right. So here's a good question. We know everyone. What's the total number of people we're needing to get vaccinated, plus everyone that has been infected and recovered? So what's the number we want to get to for vaccine and recovered? And we're sort of saying the recovered group needs to get vaccinated too, right?
Eventually, yeah.
So what's the
but
number?
even if you assume not, you assume maybe they just need one dose of the vaccine.
Yeah.
Everyone's guessing when they say this number, when they say 80% or something, 85%. That's a calculable number, but it shifts over time as immunity in the population changes, as behavior patterns change. It's somewhere up there. But the important thing to think about is you don't have to get to that 80% number before you start to see really dramatic changes in how the virus moves through the community. You can imagine if half of people were immune against the virus, for example, it's going to move more slowly. There's going to be fewer cases, therefore fewer hospitalizations and deaths. And so you don't totally wipe out the virus, as you would if you were at 95% immunity, but it's still going to provide a significant benefit.
Right. I mean, when you think of the billions and billions of replications that go on in a single individual times hundreds of millions, I mean, if you can get that down to hundreds of thousands, millions, you're making a gigantic multi orders of magnitude effect on the virus.
If you can say you've got a 100-fold lower chance of a bad mutant happening, that's a win in my book.
Yeah, and we may be talking about 100,000-fold reduction, right? Really, when you really look at the numbers here. If you like what, of course, I've not introduced formally Morgan McSweeney, and I don't know how we got Dr. Nock out of McSweeney, but it's certainly easier. And it's
try typing it in.
And if you like Dr. Nock, he's on TikTok at dr.noc. I religiously, I look for I'm following him, and I look for his videos every day because they're literally the best source of update, particularly on vaccine and. What do you consider the topics you're covering? A vaccine you kill every time, but you do a little more than that. You really talk about the good news in terms of the epidemiology as well.
Yeah, drug updates, epidemiology. Prior to this big vaccine push, I also talked about sort of more general health topics, the effects of sleep, exercise, everything like that. But now it's sort of very coronavirus focused. Anything
coming down the pike? You've mentioned you guys are producing some sort of monoclonal polyclonal antibody. Are there any therapeutics coming down the road that you're excited about or interested in watching?
Yeah, there's a couple of really cool ones. One of the coolest ones is a decoy. And so the virus binds to that receptor ACE2 on your cells. Turns out you can actually give ACE2 as a drug. And so what happens then is the virus, instead of binding to your cells, it binds to this sort of one that you've dosed, the ones that you've administered. And so it just locks on and then it gets eliminated through your mucus. That one seems pretty promising. Maybe it's a little bit harder to make than some of the others. There's some pretty early data.
Is that a systemic? Is that a systemic drug, or is that something you spray up your nose or something? How's that work?
I believe it's being tested with both. I would
Interesting.
expect both may have an impact because it actually has a second role. There's a reason you have ACE2 in your body, by the way. And it sort of has a general anti-inflammatory effect. You can think of to simplify a very complex topic. But one of the reasons you have bad COVID outcomes is you have this feed-forward cycle of inflammation. We think maybe having more ACE2 could reduce the inflammation and then also address it from that standpoint, in addition to sort of soaking up the virus.
That's interesting. And isn't ACE2 involved in right atrium renal communication on blood pressure?
Yes.
Mm-hmm. Yeah, the renin-angiotensin system, so-called.
That's the one.
And your other drug?
Yeah, inhaled corticosteroids for, one and the same, used by people with asthma, people with COPD. There's some early data, just phase 2 data, relatively small. One thing that's been very confusing, though, since the beginning of the pandemic is why don't people who have asthma, people who have COPD, you would expect them to have worse outcomes from COVID. They don't, as a general rule of thumb. And so what we're seeing now in this clinical trial where people are being treated with these inhaled corticosteroids, it appears to reduce your risk of hospitalization. That has to be obviously confirmed in bigger trials. If it is confirmed, that would be really promising. We'll have to see. There's a lot of stories like this where you have promising phase 2 clinical data and then you're sort of waiting to see if it ends up being true.
Is it only budesonide, or are they using other corticosteroids?
I think the only one I've seen is for budesonide.
Budesonide had a strange story. You know where that came from?
No.
A very good ER doctor started using it, and then gave an interview about what had happened that led him to use it. He said he prayed to God and asked God to tell him what's going to work for his poor patients, and God showed him budesonide, and he started using budesonide. And because everyone had
pharmaceutical development,
and because everyone had sort of Trump derangement syndrome at the time, anybody who said anything like that, it's like, it's the worst thing ever. He's going to kill people. And it turned out to be.
To be fair, that's not very convincing scientific evidence.
I'm just saying, it turned out to work. It turned he was using it, and it seemed to be working. And so, whatever inspired him to use it, it was inspired and had some utility. And then people started studying it formally. But yeah, I am with you. I am.
That's fascinating.
I'm looking for real evidence before we make any
certainly before I start inhaling it. I want to see.
Well, budesonide's a harmless drug. It's the same thing. It's in Rhinocort. It's just in the nasal sprays and everything. It's a nothing. But it's weird that I don't quite understand why just that one. I mean, we have more powerful inhaled corticosteroids too. I don't know why that one is the one that everyone's focused on. It seems weird to me. It's called Asmacort, I believe, in its usual form. Okay. I think I have asked I hope I haven't exhausted you with all my questions.
No, this is great.
Because I had a million of them. Let's see if there's anything else that, and by the way, the variant for the B1351, is it the E44K or E484K? The variant, is that, or am I confusing the variants?
It is. No, that's the one. It's E and then 484.
484. Yeah, 484. Yeah. Because I've heard people say that the 8 goes by so fast, I'm never sure if people are saying 44 or 44. So
yeah, and that just means, for anyone who doesn't know that nomenclature, it just sounds like random letters. It means at the 484th amino acid in that protein, it changed from one amino acid to another amino acid.
I guess we should wander a little bit into more epidemiologic area. I won't bother you too much longer, but it seems to me that a lot of the behavior of this virus is just the behavior of this virus in terms of epidemiology. In other words, it goes up and it goes down because that's the ecology of this virus. Now, humans can have an impact on it, but I have a hunch that we don't have as much of an impact on it that we'd like to wish we did. Am I off base?
I think so. I think a little bit. And so we see very strong relationships between, you can plot when restrictions start to be tightened. And then 2 or 3 weeks later, you start to see a decrease in case incidence. And then when it starts to hit this trough, looking back historically, and we release those restrictions 2 to 3 weeks later, you start to see it spike up again. And so certainly there's a lot sort of under the layer.
We had some of that. Some of those sorts of cause and effects were going on during the spring, but this latest outbreak, I can tell you for sure here in California, we had exactly the opposite. The opposite. We locked down, the thing went way up, and of course the non-scientific thing to say is if we'd locked down harder, it would have worked. Like I can't say that. All I can say, it went up and up and up and up, and then it came down right at the time that everyone said it was going to go up higher, which was Christmas and New Year's. That's when it plateaued and came down. Then they said there was going to be a Super Bowl spike, and then it accelerated down. So
Yeah, I think the tip of the iceberg, I think you're right that there's a lot that we don't see sort of beneath the surface. And a big part of that, a lot of people deny the value of herd immunity from natural infection. A lot of people think scientists deny the value of herd immunity from natural infection. That will absolutely influence the trajectory of a local outbreak.
Yeah.
If in the last couple months, you've had 30% of people get infected or something, just inherently the subsequent rate of spread is going to be lower. And some people aren't really prone to getting this virus, we think. Right. And some people aren't prone to spreading the virus. We have seen a lot of heterogeneity
Right.
where most people don't infect very many people. Other people infect a lot of people.
Yeah.
It's really hard to predict who's going to be which one of those two.
Yeah, there was a study that came out yesterday in something, one of the JAMAs, and it said essentially fat old people spread. That was their conclusion. Older and fatter. I was like, okay, that one's not going to hit the press very anytime soon.
No,
But
and tricky generalization too, because obviously there can be young people who spread a lot, and
Yeah.
it's hard to say it's only one group.
yeah, oh my God, the dog has sat on my wires here. Okay, there he is. And then I guess the final question would be schools. Do you want to wade into that territory?
Yeah, that's tricky territory. From a sort of pharmaceutical perspective, we were talking about the benefit risk sort of analysis earlier. That's how you have to look at it. And so a lot of people are very confused about, if we closed down schools back in March and April, kids weren't vaccinated then, kids aren't vaccinated now, why would we send kids back to school? One thing that has changed considerably since that time is a concern back then was kids are going to get infected. They're going to bring it home to people who are high risk, which is going to cause very bad outcomes.
That it was going to behave like influenza, which is primarily child transmitted.
Sure. Yep. We didn't really know at
Yeah.
the time.
Yeah.
Now what we do know, well, certainly the people at home who are high risk, who could be brought back the infection to, are having increasing access to vaccines. There is still absolutely risk to kids. There's no doubt about it. But there's also a risk to not having kids in school. Right? The benefits of school from sort of a developmental perspective, from a social perspective. I'm not a developmental psychologist, but
Right.
they're immense.
It's lives versus lives, right? It's COVID lives versus mental health and cognitive social development lives. It's
Right.
life trajectories and mental health outcomes versus COVID outcomes. And it's not.
You start to see a disparity across, socioeconomic impacts of learning
yes.
from home.
Oh, yes. Oh, yes. The
and
ones
so-
that they claim to want to help most are the ones they're hurting the most.
Now, one thing is there's a big difference in how safely you can structure sort of a school environment. Opening the windows makes a big difference. Wearing masks in school makes a big difference. Being
Right.
distant when you can makes a big difference. And so the difference between doing all those things and not doing all of those things could really be the difference between a relatively safe opening and a relatively unsafe opening. And so we sort of have to hold them to high standards while acknowledging there is risk, but there's also risk of not going back to school.
Right. That's right. It's lives versus lives. And by the way, the good news is the schools do a great job of keeping it safe. They do a really good job of making actually it's safer than the community at school typically. How about the CDC? Aside from the issues around the White House and messaging and stuff, how do you feel the CDC performed during all this?
Yeah, I think it's inextricably linked to the messaging from the White House. Something we kind of learned only after the fact was that there were, certain pressures on messaging and revising of documents and guidances and stuff. That being said, a lot of the communication from the CDC has sort of failed to interface with the public, how the public interprets things. There's a lot of very scientifically worded recommendations and guidances that come out from the CDC that are 100% technically correct. I read it, I understand what they've meant. Those types of things have been vastly misinterpreted over the course of the pandemic. And so I think there could have been more emphasis on, this is important epidemiological data for epidemiologists to interpret. We're going to make it publicly available. We also need to have that emphasis on making sure that we're sort of explaining the data rather than letting people interpret it. And what we saw is people interpreted to say, COVID's not a big deal. No one's dying from COVID.
Yeah.
Which is wrong.
This was the thing that I've been saying from the beginning is that I what happened to our public health officials? I was very active in the AIDS epidemic. I was way deep in that. And one of our concerns, the reason I went on radio, and just much like you going on TikTok, it motivates you to go out there and help educate. And it was Anthony Fauci that told us young people to go out on the radio and educate about a term that later be called safe sex and how HIV is transmitted and all this stuff. And I took it very much to heart, and I did it. And across the course of about 7 to 10 years, we learned how to communicate complicated epidemiological and biological material in such a way as to influence behavior. And I've said it before in these streams, it was narrative, relatable source, so a character that you can relate to, a narrative that you're interested in, humor, and music. That was it. You had all those things, and you could then shape people's behaviors. Anything else failed, especially
you
people.
can't just release a spreadsheet of data.
Yeah, especially people in a white coat talking science. It's just the worst thing in the world. They if anything, do the opposite. So I don't understand what happened. I don't understand why they just abandoned literally 40 years of knowledge about public health messaging.
Yeah, and this has honestly been a frustration. Is if you look on TikTok, you're not going to find, a CDC public health sort of messaging platform, which I don't understand why that's the case. It's mostly people, and I'm very glad that there are lots of people, other doctors, scientists, who are making these sort of volunteer-based informational pieces. That shouldn't be a volunteer effort. That should sort of be a concerted public health effort.
Completely agree with you. Or what I would advocate is you take somebody like you that's had success in reaching people through these platforms, and making sure that what you're saying is consistent with what they're seeing, and that everyone's lined up. That's it. That's all they got to do,
Right.
And get
great.
as many influencers as possible. And then, of course, the liability of TikTok is there's more than just you out there, and you're the one that has the good information, but not everybody knows that, and so that's the liability.
Vast
That's the problem.
abundance of misinformation.
Oh,
and
my
the trouble
goodness.
is, even going back to the fertility thing, there's no link between fertility concerns and vaccines for anyone who's turning in. But it takes you 2 seconds to hear someone say, "Is there a link between fertility and the vaccines?" And you're
That's right.
worried.
That's right.
It takes 5 minutes to explain the science between why that's a bogus concern and not concerning at all. And so there's that mismatch, which is really challenging to overcome.
I don't know if you're used to dealing with people clinically or not, but does it cause fertility problems or miscarriages? They walk away with that. Your 5-minute of explanation has to be gone over 7 to 10 times before they walk away with half of it. You know what I mean? That's just the way our brains are structured. Not that people are not anything against our cognitive faculties. That's how our brain works. Something dramatic and emotional we hang on to, something complicated and sort of not unfamiliar to us, it takes a while for us to get. Hang on a second, Dr. Nark, hang on. Susan's looking agitated. You okay? You're not okay?
No, I'm not okay. You know that. No.
But I mean, other than your usual not okay.
No, I was looking for a tripod because I have to set up a second camera since that other one flaked out on
got
me.
it. Okay,
for
she's
Kennedy.
worrying about the next hit here. So.
I know. I got to do another show.
Dr. Knock, you were about to comment on what I just said. Go ahead.
Yeah. What did you just say?
I talked about that people typically,
misinformation.
people typically take away 10% of what you tell them at each visit. That's what we know as clinicians. As we feel lucky when they take away 10%. And
So
if
what
you
you
if
end
you
up
say
with-
something dramatic and scary, they'll remember the emotional content.
That's exactly it. And so you get, accounts and videos posting on TikTok or Instagram. It's someone standing there in scrubs and they're saying, "This vaccine, you'll never have children and you'll lose a leg or something."
Right.
There's no data supporting it, but it goes incredibly viral, 2 million views overnight or something. I don't know what there is to be done about that. You can report it. Even if it gets taken down after a day, though, that's 2 million people who are now afraid, which is a huge number of people.
And what do they call it now? Vaccine hesitancy. Yeah, that was a word I just heard for the first time about a week ago. I'm like, ooh, I like that.
Very few people are actual anti-vaxxers.
Yes.
Flag carrying, I hate all vaccines. A lot of people want to know more. They have questions they want answered about the vaccines.
Yep. No, that's please, everybody follow Dr. Knock. That's the way we combat this thing right there. There it is. No, really, you hear you talk enough and bring the information repeatedly and reliably. Eventually, people start to listen.
It takes a while. If there's 200,
does take a while.
100 threads of misinformation, it's like whack-a-mole. You can disprove one of them. There's still 99 things that people are concerned about. And it's not hard to find new things to be concerned about when it comes to COVID vaccines.
That's exactly right. Well,
I mean, not based on evidence, but based on social media.
I feel like we covered the landscape pretty well. Is there anything I missed?
This has been great. I don't know no other topics. Appreciate you having me on. This is really good. We need to do more of this.
Okay, let's do more. We can do an update in a couple of weeks again. Believe me, I'll be updated on your daily TikToks. You're laughing. I'm not kidding. I always check in to make sure I'm not missing something. And it's funny. I mean, my son's a big TikTok. What would you call him? Influencer, user, and he was immediately knew your material, and I was delighted on both my sons. They were like, "Oh, they listen and watch." I'm like, "Good. That is good information. So keep watching." I have not been on your Instagram. Whom do you want over there? Same kind of thing.
Same thing at dr.noc. And a lot of the material is, I take my TikTok videos, I post it to Instagram, and then in addition, I'll have a story that's like, ask me anything, and I'll answer sort of, different types of questions. But the content is right now mostly COVID vaccines, COVID epidemiology, health and stuff. Very similar across the two platforms. Different groups of people on the two platforms, though.
Interesting. If there's stuff you need to push out, let us know. We'll be happy to bring you on at a moment's notice.
Can I show an example?
An example of his Instagram? Oh,
I don't know.
TikTok. You have a TikTok? This is
dear.
awesome. This is awesome. Here we go.
Should you wait for the COVID vaccines that have been updated for the new variants? The answer is definitely no, you should not wait, and this is why. It is true that the vaccines are slightly less protective against moderate COVID-19 with some of the variants. However, they still
forgot
appear
to
to do a
put that.
really, really
Right.
good
I don't
job
think
of
we've
reducing
seen
your
it.
risk of hospitalization or death from COVID-19, even with the new variants. If there is an updated version of the COVID vaccines in the future for the variants, you can get that as a booster shot. You're probably going to require a booster shot anyway, regardless. We don't know at what frequency, but eventually you probably will. But waiting until sometime in the future when there may or may not be an approval of a variant-specific vaccine is not a good idea. You should get whichever one you can right now to at least give you good protection against really severe disease, hospitalization, or death. The vaccine.
And it took him an hour to do that, everybody.
Okay.
And I remember that
You get a
one.
lot of information in there.
I know. I remember that one.
Takes time. You'll notice the same backdrop. This is the living room
I
social
know.
media special.
And do you know who Richard Diebenkorn is?
I don't.
Look him up. You'll see your painting was inspired by that, I think. So
Okay, that's good to know because my wife and I painted it, and we had looked up some abstract art online, and then we just sort of
You copied
painted
it?
I'm sure that's you. You saw his stuff. He's a very common.
Wait, you painted that behind you?
Yeah, my wife and I. I'll
Oh,
kind of.
let me see. Oh, that's
you go.
adorable. Look at
Yeah,
that.
I dig it.
We have
I
a
dig it.
fan who's an artist. Wiz Chris will like that.
She was on the thread a minute ago. Let's see if she's in there.
Yeah, she'll like it.
Okay. Last questions here.
Okay.
Any opinion about vitamins, vitamin D, zinc, quercetin, all that stuff?
Yeah, there is pretty good evidence that if you're deficient in vitamin D, you may have increased risk of more severe outcomes. It doesn't change your risk of getting infected in the first place, but if you do get infected, you may have more severe disease.
And let me just stop you right there. And I've heard this exact point of view, but I have to tell you, clinically, when people have low vitamin D levels, even they're not, let's say they're low, like I think 30 or 35 is the lowest lower limit of normal, and they may be 37, which is very, very common for people to have levels in that zone, particularly as you age. It's really hard to get them up. It's very hard. That's why we have prescription strength vitamin D. But go ahead, finish your point.
Right.
Yep.
Yeah, no, that was it. If you're deficient, you want to be not deficient, probably.
Yeah.
An important point from my dermatologist friends is that the best way to get that vitamin D up is not go lay in the sun. You can get equally good source of vitamin D from food, from supplements. If you're in the sun, you should be wearing sunscreen.
You should wear sunscreen. You got to convert to 125 dihydroxy vitamin D, which is UV that does that conversion. So you have to
Yeah.
have some sunlight.
Yeah.
No, you
Zinc.
still.
It's yeah.
I like vitamin D
Zinc.
from the
Anything
sun.
zinc or quercetin? Do you think that's doing anything?
I don't know enough to
Yeah.
respectably comment.
I'm backing off the zinc now that I'm post-COVID. I'm convinced that I've read a lot of the material on zinc being useful for immune functioning. The question is how much, how long. I don't know. We just don't know that. But I'd be careful. It affects.
Ivermectin is another one.
Ivermectin.
Not very good evidence that ivermectin is useful either as a prophylactic for prevention or a treatment for treatment. Probably the best evidence for this is you can go talk to Merck, who makes an ivermectin-based drug, and they've got a page that says there's no evidence that our product should be used prophylactically or therapeutically. That being said, those trials are ongoing, so we will eventually have an answer. But right now, totally speculative. A lot of people claim this sort of, if we have this, why do we need vaccines? Because we don't have this. There's no data supporting
Right.
the use of
That's
ivermectin
right.
in that way.
That's right. It's we're trying to do anything we can to give ourselves a hedge to, either prevent the infection or prevent from going to more severe disease. And I understand why we've all reaching for these manipulations, so to speak, of our immune function. I wouldn't discourage it. I've encouraged it all along. But, once, like myself, once you've had the vaccine, once you've had COVID, now I start to worry about copper metabolism, which is affected by zinc. I don't know about long-term quercetin. We just don't know. So now we're getting into a zone where it's like the risks may not be outweighed by the potential benefits. So we're in an interesting zone here.
Yeah.
All right. We're going to have to wrap this up here. Susan, I see Susan trying to prepare for the next thing she and I have to do.
Okay, Dr. Drew, it's been great.
The privilege is mine. Everyone is very excited, including myself, to talk to you. You did not disappoint. I feel like we covered. I'm sure, though.
And Drew booked you all by himself.
No, I was very pleased. I'm
The
really,
fan.
Yeah, I am a fan, and I'm probably going to walk away and think of 10 more questions I want to ask you. So that's good. That means we'll have to bring you
doubt.
back. So, and no doubt.
But tell your
down.
9 million followers to wait a little bit till I worked out the glitches in the video at the beginning because it's not going to look good. It's everybody who
We'll give
knows
ourselves a little buffer.
everyone who knows us knows that it's funny that I screw everything up every time. So
Well, today was a bad day.
Worked
for you.
out well.
Yeah, worked out fine.
I like the new angle. I think we're going to use this for the
Good.
future.
Dr. McSweeney and myself are fine with this. We got to do what we wanted to do. And I think we got a lot of good information out. Those of you on the Restream, we thank you very much.
I'm going to play banners on the way out instead of ads because I couldn't get the sound to work. So, and I don't even know if you can hear the music. So here we go.
Oh, well, hang on a second. Already people are talking about, because you work in biotech and they're already starting to take aim at pharmaceutical companies. Oh my God, they're motivated. Blah, 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 their motivation.
The reverse of motivation.
Go ahead.
I work in the development of treatments for COVID. Okay. So maybe you can say I have a bias in the use of monoclonal antibodies. But what you have to understand is there's a reason why I'm supporting vaccines, because I think they're good for public health. If the vaccines worked really well, and they do work really well, that inherently means we're going to sell less of our drug. You don't need as much of a treatment if you've got vaccines.
Yeah.
And so the only reason I do this is to promote public health. I don't get paid by, anyone to do it. It's just sort of my little endeavor.
Yeah. If we cannot be as a medical system be trusted to advocate on behalf of the health of the public, we got a bigger problem on our hands here. So just be careful with your paranoia. We have some weird collective delusion going on in this country right now where everyone's seeing Nazis, everyone's paranoid of drug. I've never seen anything like this. Literally, if you'd come to me with these feelings about seeing Nazis and worrying that you're being drugged or poisoned by a drug company, I would have put you in a hospital 4 years ago. Now that's the general tone of the public. It's uncanny how wild we've become.
I will say, though, keep it up. You should keep questioning the people you're getting your information from.
For sure.
Including me. That's why I cite my sources. A lot of people don't cite their sources. You should call them out.
100%. And so understand there's a difference between asking people to cite their sources and questioning them versus being paranoid about them. There's two different postures. So that's all I'm advocating for.
You're getting nothing but love on our Restream.
All right. Excellent. Okay. So Morgan McSweeney, Dr. Knock is where you can find him, at Dr. Knock. We will hopefully talk to you again soon.
Sounds good. Dr. Drew, it's been a pleasure.
Thank you so much.
Okay. See ya.
Bye-bye. And we'll see you all tomorrow.
Okay.